about to show you how paleontologists solve a surprisingly tricky puzzle, telling apart animals that at first glance look the same. When you look at theropod dinosaurs, they seem remarkably similar. Bipedal predators with sharp teeth and long tails. But scientists have developed a forensic approach that examines hundreds of anatomical clues. Every feature tells part of the story, and when combined, these details reveal not just what these animals were, but how they lived, what they ate, and how they're connected to the birds flying outside your window right now, as recent tooth and skull studies show. How can a tiny clue in a mystery tooth tell scientists it belonged to a predator no one expected in Morocco? The answer starts with understanding how paleontologists read the most important evidence of all. Picture two theropod skulls sitting side by side on a laboratory table. To most people, they look nearly identical: elongated snouts, rows of sharp tooth sockets, and large eye openings. But to a paleontologist, these skulls might as well be carrying name tags. One could belong to a massive Allosaurus that terrorized Jurassic landscapes, while the other might be from a smaller Ceratosaurus that lived in the same time and place. The differences are they're hidden in plain sight. The skull serves as nature's most reliable identification system for theropods. While body size and overall shape can vary dramatically within the same species, skull proportions remain remarkably consistent. Scientists treat each skull like evidence in a forensic case where small differences in measurements and ratios, sometimes only a few millimeters in a bone, can be diagnostic when combined with other features. The distance between eye sockets, the angle of the nasal cavity, and the positioning of skull openings, called fenestrae, create unique patterns that separate entire evolutionary lineages. This precision reaches extraordinary levels. The shape of the antorbital fenestra, the large opening in front of the eye socket, varies dramatically between groups. The antorbital and temporal fenestrae vary by clade and can help separate major families. When combined with other skull measurements, they are powerful indicators. Paleontologists approach skull analysis like forensic investigators examining evidence. They focus on key comparisons. Orbital diameter versus skull length ratios help align species, while the shape of antorbital fenestrae helps separate groups. These ratios reveal evolutionary relationships that occurred across vast stretches of time. Recent morphometric work, including studies this year, shows skull shape links to both family relationships and feeding style, letting paleontologists read ecology from bone geometry. Carnotaurus skull and neck anatomy suggests feeding specializations different from other large theropods. Paired with tiny arms, this signals a different hunting style, while a Spinosaurus skull displays elongated proportions, perfect for catching fish. What makes skull morphology so powerful is that it encodes entire evolutionary histories in its architecture. Every curve, angle, and opening represents millions of years of adaptation and survival. The skull doesn't just identify the species, it reveals how the animal lived and where it fits in the grand theropod family tree. But sometimes a single fragment tells an even more dramatic story. A single tooth fragment no bigger than your thumb can reveal whether a massive predator spent its days hunting fish in rivers or tearing apart other dinosaurs on land. The answer lies in microscopic details that scientists have only recently learned to decode properly. These teeth sat in museum drawers for decades, defying classification. The chem beds in Morocco had yielded thousands of isolated theropod teeth over decades, creating one of paleontology's most frustrating puzzles. These fossil deposits contained a jumbled mix of predator remains from the Cretaceous period, and scientists struggled to match isolated teeth with their original owners. Some experts remained convinced that certain sharp, curved teeth belonged to raptor-like predators, but the evidence never quite fit. A 2024 study of isolated teeth from the Cenomanian chem beds applied a combined approach using cladistic analysis, discriminant analysis, and machine learning algorithms. The breakthrough came when scientists realized that serration patterns act like biological fingerprints when combined with other data. Flesh-tearing theropods developed distinct zigzag serrations along their tooth edges, creating microscopic cutting tools perfect for slicing through muscle and bone. These denticles vary in size, shape, and density depending on the prey type. Fish-catching specialists like Spinosaurus evolved completely different dental equipment: smooth conical teeth designed for piercing and holding slippery aquatic prey rather than cutting. Serration patterns, size, shape, and spacing act like a fingerprint when combined with other data, and modern methods, including machine learning, can match isolated teeth to likely groups. The spacing between serrations, their triangular versus rounded shapes, and their distribution patterns created unique signatures for different lineages. The mysterious chem-chem teeth finally revealed their secrets through this advanced computational analysis. Those supposed raptor teeth actually belonged to abelisaurids and noasaurids rather than dromaeosaurids, completely unexpected families that had evolved convergent tooth shapes for similar hunting strategies. Recent dromaeosaur skull studies and analyses of Spinosaur skulls show how tooth and skull form can overturn simple predator stereotypes. Serrated teeth instantly identify predators that attacked large prey, requiring cutting tools to process their meals. Smooth teeth reveal specialists adapted for catching fish, small animals, or processing softer foods. This dental detective work provides immediate insights into ancient food webs, but teeth aren't the whole story. Sometimes the most shocking evolutionary secrets hide in the most unexpected places. The most dramatic evolutionary betrayals show up in theropod limbs, where massive predators developed completely contradictory anatomical strategies. Carnotaurus had dramatically reduced arms. That extreme reduction, combined with its skull and body plan, suggests the forelimbs played little role in feeding. Interpretations of its hunting style remain debated. Meanwhile, Spinosaurus developed powerful forelimbs with enormous claws perfectly designed for snatching fish from rivers. This bizarre contradiction reveals one of the most important classification tools paleontologists use. Finger count alone tells the evolutionary story of theropods across millions of years. Primitive forms like Calophysis retained 4 functional fingers, maintaining the ancestral pattern inherited from earlier archosaurs. As theropods evolved, they systematically lost digits, a common trend in many lineages, though not uniform across all groups. Allosaurus and its relatives reduced to 3 fingers, while the mighty Tyrannosaurus rex ended up with just 2. This digital countdown marks major evolutionary transitions and helps scientists place species on the theropod family tree. The contrast between arm lengths reveals specialized hunting strategies. Tiny arms combined with skull features indicate one strategy, as seen in Carnotaurus and other abelisaurids, that relied on powerful leg muscles and skull-based attacks. Long, powerful forelimbs with claws indicate another approach, as Spinosaurids developed fishing equipment perfectly adapted for river systems. Pelvic architecture provides equally dramatic evolutionary fingerprints. Changes in the pelvis affect posture and locomotion, and are used in phylogenetic studies that combine many traits to place species into families. Early theropods possessed relatively simple pelvic structures, but as different lineages adapted to specific ecological niches, their hip bones underwent radical transformations affecting balance, stride, length, and overall locomotion patterns. Environmental pressures throughout the Mesozoic drove these anatomical innovations. Rising sea levels created new aquatic hunting opportunities, while changing vegetation patterns favored different terrestrial predation strategies. For example, newly described Alp Karakush from Kyrgyzstan was placed in Metriacanthosauridae, largely on pelvic and hind limb characters, a reminder that hips and limb bones can be decisive. These limb proportions and pelvic shapes function as evolutionary signatures, instantly revealing both hunting methods and phylogenetic relationships. But beyond arms and legs, other anatomical features tell even more sophisticated stories about ancient behavior and survival. Tail anatomy reveals some of the most sophisticated engineering solutions in theropod evolution. Some dromaeosaurs had stiffening structures, including ossified tendons that made their tails more rigid and useful as balance or maneuvering aids. Velociraptor and its relatives transformed flexible tails into precision steering mechanisms, allowing agile hunters to execute sharp turns while pursuing prey at full speed. Other theropods possess standard flexible tails that couldn't provide this level of control during complex maneuvers. Evolution produced some truly bizarre ornamental features across theropod lineages, raising questions about their purpose. Ceratosaurus sported a prominent nasal horn, Dilophosaurus developed paired crests along its skull, and Spinosaurus carried a massive dorsal sail that could reach 6 feet in height. These ornaments are useful identification markers, especially when combined with other anatomical data. Each ornamental feature serves multiple purposes beyond simple identification. These structures were likely used in display and recognition, but their precise roles are debated. They are still valuable taxonomic markers. The elaborate headgear helped individuals recognize their own species in environments where multiple theropod species coexisted, revealing complex social behaviors that scientists are only beginning to understand. The feather revolution transformed our understanding of theropod classification entirely. Fossils preserving feathers, especially among Maniraptoran theropods, show a progression from filament-like structures to pennicheous flight feathers, directly linking many theropods to birds. Preserved plumage creates a direct bridge between ancient predators and modern birds, showing evolutionary connections that seemed impossible just decades ago. Integumentary features provide crucial classification data that extends far beyond basic identification. Scientists can trace evolutionary development through preserved feather types, from primitive proto-feathers to fully developed flight apparatus. These discoveries connect diverse theropod lineages through shared ancestral characteristics. Combined with dating and cladistic comparisons of many traits, these anatomical details let scientists build robust family trees. Every skull, measurement tooth, serration, limb proportion, and integumentary feature contributes essential data points across hundreds of species. That is how bone, tooth, feather, and rock come together to tell who belonged to what family, and how some of them evolved into today's birds. Modern birds are the surviving theropods, carrying forward the evolutionary legacy of these ancient predators through unbroken lineages. The anatomical detective methods we've explored create a diagnostic pipeline. Skull shapes and fenestrae point to lineage relationships. Teeth reveal diet and can be matched using modern statistical methods like the chem, tooth study, limb and pelvis anatomy show locomotion patterns and phylogeny, while feathers and integument create direct links to birds. Next time you see a sparrow, remember it carries echoes of those ancient hunters. The same skeletal architecture that supported massive Cretaceous predators now powers modern flight. Paleontology is a detective story written in bone, and it keeps rewriting itself as new methods and fossils turn mystery into detail. Which anatomical clue surprised you most? Tell us in the comments. And if you enjoyed this deep dive, subscribe for more evidence-driven dinosaur stories.